Hello everyone, welcome to Manocha Academy. In today's video, we are going to perform a few biochemical tests to detect the presence of certain substances like protein, glucose, sucrose, starch, etc. in the kind of food that we eat. So we will be taking some natural samples and we will be testing them for the presence of glucose, sucrose, proteins, fat, etc. and we'll see what kind of tests we'll perform and what are the observations that we are supposed to get in case those substances are present in our food. So let's get started with the experiment. The first experiment that we are going to perform is for the detection of glucose. Now for that I have taken banana extract. So I have mashed the banana prepared with this extract. In the laboratory you will be provided with a banana extract, and I am going to test it for the presence of glucose with the help of this reagent which is known as Benedict's reagent. What are the things that I need for this? I need test tubes, I need a spirit lamp, Benedict's reagent, the sample, that is a food sample. You can take the food sample from any other kind of food to test the presence of glucose, but if you want glucose to be present and show a positive result, then it's better that you take food which is rich in glucose. And I will be needing a test tube holder. So let's get started with the experiment. For this, I will first take a little bit of the sample in the test tube. So I've taken some sample in the test tube. And now I will be adding Benedict's reagent to this with the help of a dropper. As you can see, I just added 5-6 drops of Benedict's reagent, and as a result of which the entire solution has turned blue, which is the color of the Benedict's reagent. Now what I will do is I will heat it, and on heating we will see that a brick red precipitate is formed if glucose is present. Now I will be heating this solution that I have made of glucose and Benedict's reagent, and you will see a color change. You see already the color has started changing. It is initially slightly greenish in color, and then it turns yellow. And slowly you will see that the color is changing further to almost brick red. As you can see, the color is changing, and this is confirming that glucose is present in the food sample that we have taken. Okay, so this is the first test to confirm that glucose is present in the food sample. We'll perform another test to confirm the presence of glucose, and we'll see what that test is. So the next test we are going to perform is called Fehling's test. In this test, for this test, we will be requiring Fehling solution A, which is here Fehling solution 1, and Fehling solution B, which is here Fehling solution 2, and we'll be needing the food sample which has glucose in it, and of course we'll be needing our test tube holder, a dropper, and we will be needing the spirit lamp. So let's start with the experiment. First we will have to take the solution, the food sample that is, in a test tube. To this I will be adding Fehling solution A first. Just a few drops, 5 to 6 drops. And so it should be equal amounts. If you are taking 2 ml of the sample, then 2 ml of Fehling A and 2 ml of Fehling B. And then Fehling solution B. Oops. As you can see, the color has already changed, but we will look for further change in color. So we will be heating it, and if glucose is present in the sample, then we will be getting a green, yellow, orange and brick red color. So let's see if this color changes. You already notice a color change here. So if the amount of glucose present is sparing, it's just traces of glucose, then it should be greenish or yellowish in color. And if the amount of glucose is more, it should be orange or brick red in color. So depending on the amount or the quantity of glucose present in the food, the color also varies. So as you can see, the color has already changed to orange and almost reaching brick red. So there you go, the brick red color has already appeared, and this confirms the presence of glucose in the sample of food that we have taken. Okay, so these are the two tests for glucose. Now we will move on to the next test, which is the test for sucrose. That is another carbohydrate, and we will see how we can detect the presence of sucrose in our food. So the next test that we will be performing is to test the presence of sucrose in our food. Now sucrose, as you know, is a disaccharide, and it is made up of glucose, one molecule of glucose and one molecule of fructose. So what we are going to do is we are going to hydrolyze, that is break down this sucrose into glucose and fructose and do the test for glucose again. So for that I have taken sugar solution. Sugar, as you know, contains sucrose, so I have taken sugar solution. I have taken sodium hydroxide 40%, I have taken concentrated hydrochloric acid, and I have taken Benedict's reagent. I will be needing some test tubes, my spirit lamp, a test tube holder and a dropper for this. So first and foremost, the first thing that we need to do is we need to break down the sucrose into glucose, a process called hydrolysis, and for this I will be taking some sample, food sample in the test tube, and I will be adding hydrochloric acid, concentrated hydrochloric acid. So we need to be very careful. I have taken some concentrated hydrochloric acid, a few drops, and adding this hydrochloric acid, we are trying to hydrolyze, that is break down the sucrose into glucose and fructose. As you know, in our stomach there is hydrochloric acid, and this hydrochloric acid is also responsible for hydrolysis, that is breakdown of sucrose into glucose and fructose. So I will do this, and I will quickly put the cap on my hydrochloric acid bottle so that we are all safe here. Now I will have to make this solution neutral, alkaline actually. So for that I will be adding a base, and I am taking 40% sodium hydroxide solution. I am taking sodium hydroxide solution, and I am going to heat it so that the process of hydrolysis is completed. You can heat it a little bit. Now I will be adding Benedict's reagent. So once this is heated up a little bit, I will be adding Benedict's reagent. So added Benedict's reagent here. As expected, the solution is blue. Now we will see if hydrolysis has occurred, which you can already see because the solution was warm enough. You saw that the color quickly changed because of the presence of glucose, and if you heat it further, you will see the brick red color. So that confirms the presence of sucrose. Okay, so the chemistry behind it is that we have taken hydrochloric acid to hydrolyze sucrose into glucose and fructose, and then we neutralize the solution with the help of a base, that is sodium hydroxide, and to that you can heat it a little bit. To that you add Benedict's reagent, and when you heat that solution after adding Benedict's reagent, if it turns yellow and then orange and then brick red, it confirms the presence of glucose. If it is yellow, it's less amount of glucose. If it is orange, it's quite a lot of glucose, and as you can see, this one has already turned brick red. So this confirms the presence of high amount of glucose in the sample. In this case, glucose means that it has come from sucrose because sucrose was the sample that we have taken. So sucrose has been broken down into glucose, and therefore this brick red color, as you can see, almost brownish, is appearing. Okay, so that confirms the presence of sucrose in the sample. Now we move on to another carbohydrate, which is a complex carbohydrate, and it's a polysaccharide, and that is starch. So this experiment is for detecting the presence of starch. For this, what I have done is I have taken a potato and I have taken a potato extract from here. For that, I have just scarred the potato a little bit, and if you just squeeze it like a lemon after scarring it, you see the extract is coming out of it. So I have taken the potato extract like this. Now what I am going to do is, I don't know if starch is present here or not. So a very easy test for starch is the starch iodide test. So I will be taking iodine, which normally is brown in color. I'll take it with a dropper. You can see the color of iodine. It is brownish in color. And as I add it to starch, it is supposed to react with starch and form starch iodide, which is purplish black in color or bluish purple in color. So let's see if that color change happens. So you can see the color here. The color is brown, right, of iodine, and now we'll see the color change here. You can see a bluish purple color has appeared. It is very clear that it is bluish purple, and it is very different from the color of iodine. If you are not very sure about the bluish purple color, what you can simply do is add a little bit of water. I'll just to show you the bluish purple color, I'm adding a little bit of water. Now you can see the color, it is bluish purple. And this is because of the presence of starch in it, which reacts with iodine to form starch iodide. As you know that the structure of starch is pretty branched, inside which the iodine can go and attach to form the starch iodide structure. So this is a confirmatory test for the presence of starch in the food sample. So those were the different carbohydrates. Now we move on to fat. We'll see how we can detect the presence of fat or oil in different types of food that we have. I have chosen peanut for this, but you can of course carry on this experiment with any other kind of oil or food that contains oil. So to test the presence of oil present in food, I have taken some peanuts. These are raw peanuts. And what I'm going to do is, peanut is supposed to contain peanut oil. It's supposed to contain oil in it. So I'm going to just crush it and press it against a paper. And you know what happens when you touch a paper with some oil? The paper turns translucent. So we'll see if that happens. So for this, I've taken some peanuts and I will just fold the paper. You use your mortar and pestle for this. I'm using a hammer because I have one handy right now. I will simply crush it. So I have crushed this and I'll now press the paper very nicely against this powder that I have made inside. The peanut has been crushed, so I am just pressing it, the paper hard enough. And now if you look at the paper, this has developed some translucent spots. And these translucent spots are because there was oil present in this peanut. As a result, the oil has made the paper translucent. So this is the confirmatory test for the presence of oil or fat in food. In this test, we are going to show the presence of protein in food. So for that, we have taken a sample of food. You can take it from egg albumin, you can take it from any other source which contains protein. So I have taken an unknown sample. So this sample contains something. We'll see what, if at all protein is present or not. And for the test, we will be needing 40% sodium hydroxide solution, 1% copper sulfate solution, and that's it. Okay, so this test is known as the Biuret test. For this, we will first take a test tube and we will take some amount of the sample that we have. Not so much, that is not essential, yeah. So this is enough, we'll take some amount of sample and then we'll add a small amount of sodium hydroxide solution to the sample. So I'm adding about 5-6, let's say about 1 ml of sodium hydroxide solution to the sample, and I shake it well. As you can see, it is colorless. After shaking it well, I will just take a few drops of copper sulfate. As you can see, the copper sulfate is blue in color. Now we'll add and we'll see what color we get here. Let's see what color we get here. We will shake it gently and you are supposed to get this purple color if protein is present in the food. There's no reason why you should get confused between the color of copper sulfate and the color of the solution after it has been added to the sample of food, because this is purple in color, this is blue in color. So when you add copper sulfate, few drops of copper sulfate to the solution, if it contains protein, then you will see that the color turns purple. And this confirms the presence of protein in your sample, food sample that you have taken. This is known as the Biuret test. We will perform the next step, that is the Xanthoproteic test, to show, to confirm further that protein is present in a sample. So in this test, which is called the Xanthoproteic test, we are going to take the sample in a test tube. We will be needing a spirit lamp, we will be needing concentrated nitric acid, and we will be needing ammonia solution, which is basically ammonium hydroxide. Okay, we have to be very careful with all these reagents because they are extremely corrosive. So we have to be careful. We should be wearing our gloves and all the lab coats and safety gear that you need. So I have taken the test tube which contains the sample. Now to this I will be adding some 5 to 6 drops of concentrated nitric acid. Very carefully. Stir it, shake it, and then I'll hold it with a test tube holder and I will just heat it on a flame for about 2 minutes. As I heat it, you will be able to see that a yellow precipitate is appearing in the test tube. So as you can see, the precipitate has turned yellow. Now to this I will be adding ammonium hydroxide solution, and we expect that this yellow precipitate will now change its color. Let's see what color change happens here. As you can see, the color has changed and it has become dark and orangish in color. This confirms the presence of protein in the food sample that we have taken. So this is the second test for protein, which is the Xanthoproteic test, and it requires corrosive reagents, so you have to be very careful while performing this experiment. So the next test that we are going to perform is Milon's test. For this we will need very simple reagents. We will need Milon's reagent, we will need the food sample and a test tube. So I will be taking a small amount of the food sample in the test tube, and to this I will be adding a few drops of Milon's reagent and just wait for some time for the color to change. We should be able to observe a pink color if there is protein present in it. As you can see, a very light pinkish color appears after some time, and this confirms the presence of protein in the food sample. So those were the tests confirming the presence of different types of organic matter, protein, carbohydrate and fat present in our food. Now here is a homework question for you. Can you tell me, if I give you a paste of peanuts, what are the different food types for which you are going to get a positive test? Please write your answers in the comments below as I eagerly wait to read your answers, and I promise to reply to all of them. I hope you enjoyed the video and learned from it. Do check out the courses on our website manochaacademy.com and our Android app Manocha Academy. Links are given below. We have courses on physics, chemistry, mathematics, biology, coding and artificial intelligence. In these courses, you will get concept videos, live classes, quizzes, mock tests and revision notes. So they will be perfect for your exam preparations. Please hit the like button and share this video with your friends. If you haven't subscribed to our YouTube channel yet, go ahead and smash that subscribe button right now and click on the notification bell so that you do not miss out on any of our videos. Stay connected with Manocha Academy and let's keep learning together.